The roundtable that Susan Dexter has put together for us includes three panelists, distinguished panelists: Joe Figlio, who is the Senior Project Officer at HHS, ASPR, and Barda; Debashis Sahoo, Director of Engineering at madeincell; and Victor Lee, who is the Vice President of Contract Manufacturing Operations at Biotechnique. Again, they're going to look at fill and finish manufacturing strategies and really taking a look at if the technologies are better or if they're just new. So we'll have some mixed perspectives and some nice information flowing back and forth. If you have questions, there will be a microphone in the audience. I will hand it to you. Please make sure that the panelists can hear you. And with that, let me introduce Susan Dexter, who is going to be your moderator and chairperson. Susan is the principal consultant at Latham Biopharm Group. So without further ado, here's Susan.
Thank you for coming. Can everyone hear me okay? I'm a little short for this microphone here. Right, so we're going to be talking about technologies for fill finish, and we have long conversations in the industry about just because it's new, does that make it better? And is it worth pursuing new, or is novel a better approach? So what we're talking about here is the complexity of fill finish. It's the last step before you get into patients. It's the most important critical step to maintain drug quality and drug of the product prior to injection. So it's a last step before it sees humans, and that's quite an important aspect of what the regulatory authorities are constantly trying to assess when they inspect facilities. The washing, depyrogenation, and filling and capping processes, although it doesn't take long to do a fill, are very important and extremely complicated systems requiring a lot of moving parts. The FDA has said that they're very interested in novel technologies and new ways of doing things that are disruptive, and want to de-risk, if you will, the GMP envelope around fill finish by introducing disposable technologies, which they've admitted has de-risked the GMP validation space around bioprocessing for drug substance, and is there a way to do that for drug product as well? So today we're going to be entertaining discussions from these three panelists to better understand kind of the existing technologies and why they're still applicable, why there hasn't been that much change in the industry over the last 30 years, and then the sequential novel emergence of new technologies for fill finish, all the way through to what one might call disruptive technology that could transform the way we think about manufacturing drug product. So our first speaker is Joe Figlio. He's got an MBA. He's a Barda project officer, and he's for the Manufacturing Facilities and Engineering Division of Biomedical Advanced Research and Development Authority, aka Barda. And the office is the Assistant Secretary for Preparedness and Response in the U.S. Department of Health and Human Services. Joe joined the Manufacturing Facility and Engineering Group in August of 2009. As a project officer, Joe monitors and reviews infrastructure-related contracts that will support domestic vaccine manufacturing in the event of a declared pandemic, including 2 Centers for Innovation and Advanced Development and Manufacturing and the projects for Fill-Finish Manufacturing Network. Joe previously worked for Merck & Company in the West Point facility for 19 years in various roles, supporting vaccine manufacturing, and at Mitos Technologies in Phoenixville, Pennsylvania, as an operational leader. Without further ado, here's Joe.
Thank you, Susan. Can you hear me okay? Speak louder? Okay. How about now? All right. Plenty of seats up front in case for all those people you standing in the back, come on down. No, actually, if you're looking for a discussion on new technologies in the filling area, you're going to be more interested in the other two co-presenters here. I'm really talking to you about an approach that the government is taking to increase our ability to fill vaccines in a pandemic situation. So more of a novel approach than it is new technology. So as Susan mentioned, I'm from BARDA, which is the Biomedical Advanced Research and Development Authority in Washington. And the next acronym I'm going to throw at you is the FEMC, which is the Public Health Emergency Medical Countermeasures Enterprise Review. A lot of what BARDA does is Are you talking to me? A lot of what BARDA does is driven by either a Congress or a presidential action or something like that. The FEMC here is one thing that BARDA listens to and responds to, and a lot of our work is created from that. So here, if you notice from August 2010, there was a need that was discovered for manufacturing capacity. The nation currently lacks the domestic manufacturing capacity to rapidly produce and package a vaccine for the American public in the face of a pandemic. This difficulty in producing, filling, and finishing enough vaccine that could broadly be available before the virus had spread widely is an important reminder of the unmet need for this capacity. So what BARDA has done over a number of years is already increase the capacity to produce more vaccines in bulk. Now we're kind of playing catch-up with the filling and finishing operation. So on the left here of this next slide, BARDA is kind of set up with core services. You can see, it might be a little small to read, but we have the Centers for Innovation and Advanced Development and Manufacturing at the top. Down the bottom right, clinical services, non-clinical network. And so they're set up to kind of mirror a pharmaceutical company with different functions, a regulatory services group. Okay, so as part of that, on the orange block there is the fill-finish network, which was established in 2013. And if you follow that arrow over to the right, those are some of the goals of the fill-finish network. Supporting the FDA, for instance, is one we've already started to do. Increasing capacity at some of our CIADMs who may not have the ability to do the filling on their own. So there's various needs that were determined that could be met by this fill-finish program. So as far as the actual objectives and capabilities, the objective of the program to establish a network of existing facilities that are pre-qualified and under contract to fill and finish vaccines for the government. Nothing new and novel in that statement. We want companies that have done this before, who are capable of doing it. We want to contract with them so we have the ability to do this in the future and at a rapid pace in the event of a pandemic. As far as some of the expertise on the right, the capabilities, obviously formulation, filling, and finishing is the obvious one of vials and syringes, but that also includes tech transfers, regulatory support, analytical development, clinical trials, and labeling and packaging, and everything else that comes with the filling and finishing operation. So we were looking for all the skills necessary to do that. You may think a CMO is obviously going to be one person that we would look at to fill that need, but it did not have to come from a CMO. So how does the government go about doing this? We issue an RFP, goes out on the street on FedBizOps. Some of the mandatory criteria that we were looking for. Number one, a U.S.-based filling capacity. So not everything BARDA does is U.S.-based. We work with some international companies, but in this case, we wanted it to be a U.S.-based capacity. In the event of a pandemic, you could have borders that get closed and other things that can't happen internationally, so we wanted the capacity in the U.S. Number two, for mandatory criteria, written evidence that the offerer has filled a license-approved product before. So we did not want someone who just came to us and said, yeah, we could do this, but hasn't done it before. So we wanted a proven capability of being able to fill a licensed vaccine product. And the third criteria, a written capability statement telling us how they were going to do it, where they were going to do it, if they need additional resources or modifications to any of their lines, their filling lines, things like that. So a complete write-up of how this will get accomplished. As well as at the bottom you can see maximum time frame, how many doses could they produce in 12 weeks once they were provided with the bulk vaccine. Okay. So general expectations from the program. First would be the tech transfer and influenza vaccine from the manufacturer within the first 3 years of award. And we're about halfway through that time frame right now. That activity is in progress on all of them. Core services contracts for CBRN projects. That's our Chem Bio division. availability for public health emergency contracts such as Ebola, and we've actually have seen some of that already take place. We have some task orders that we issued to support the Ebola efforts that's going on within the government. And multiple projects could be ongoing at the same time. And that's true with any of the CMOs. They have the capability of doing that. Okay. So the offers were made to 4 companies. Some of them may be familiar to you. First one is Cook Pharmica in Indiana. They have partnered up with GSK as their vaccine partner. They've established that agreement, and they are into the next phase where they are starting the tech transfer process. Then we have Feeon in North Carolina. They are still working on their influenza partner at this time, so they haven't moved into the tech transfer step yet. Nano Therapeutics has partnered with Dexter in Indiana also. They have their agreement in place, and they are working on the tech transfer stage now. And then PAR up in Michigan. They are also still working on their agreement with their influenza manufacturer. And here's just a geographical breakdown of where the companies are located. Nano Therapeutics down in Florida, Feeon in North Carolina, the others in Indiana, and PAR in Michigan. Now, the 2 that have not yet completed their agreement in place, there's been some other outside factors that have caused that, such as acquisitions and mergers of companies going in the past couple of years that have occurred that have kind of slowed that process down. So looking at the path for these 4 network companies for the future, first being confirming their influenza partner. 2 of the 4 have done that already, and 2 of the 4 are still working on that. They're getting closer. Tech transferring the pandemic candidate into the facility, onto their filling line, and then establishing the surge capacity, whether that's through staffing or other means to be able to staff up, and then also supporting any CBRN or emerging disease projects that Barda may have. Some gaps. We do have a few gaps in the program, but they weren't necessarily not thought out ahead of time. Live virus capabilities are fairly limited. We are trying to fill that gap in other ways, whether it's through the network or through outside the network. Sterile bag technologies is also one that we do not have a lot of capability in the network for. There's been some shortages with the FDA, that such as saline and others, that are being worked on separately. But those were not, that's not how you're basically going to fill and respond to a pandemic. We wanted to make sure we had vials and syringes, the proven technology that's able to be rapidly employed. And then additionally, as I mentioned, we weren't looking at new technologies with this effort. Barda does do that in other avenues, but not in this particular effort. We wanted to rely on proven capabilities that existed and utilize capacity that already existed, and somehow harness that through the contracts with these companies so that we could respond and fill that gap that you saw on the earlier slide of how to respond to a pandemic. We didn't want to have all this bulk laying around that we could not fill and get to the public. So it's more of a novel approach as opposed to new technology. But I'm sure as I said, my co-presenters here will have some more interesting topics to discuss there. And that's all I have for you. Any questions? No? Thank you.
We can also take questions at the end if anyone has any further questions for Joe. Our next speaker is Victor Lee. He's the Vice President of Contract Manufacturing Operations at Biotechnique and has over 20 years of broad experience in pharmaceutical manufacturing operations, cytotoxic fill and finish, product and technology development, and pharmaceutical equipment and facilities. Dr. Lee has provided outsourcing services commonly needed by pharmaceutical and biotech companies, including project management, process engineering, automation equipment and facility design, and commissioning and qualification of facilities. He's worked in various technical capacities in both large and small volume parenteral manufacturing facilities for companies such as McGaw, B. Braun, Gencia, Secor, and Teva. Mr. Lee has experience with sterile fill finish equipment and operations for both vials and flexible containers, with isolators in traditional cleanroom environments. He'll be talking about the capabilities and the new technologies being employed by Biotechniques.
Thank you, Susan. Good morning, everyone. You guys can hear me okay? Good. Okay, I'm here to give everyone an overview of the sterile liquid filling processes and technologies, specifically something called shrinking the fill zone, okay? And I'll get to it in the presentation. So here are some things I'm going to be talking about, some product considerations, container closures, the types of technologies, filling environment, and the regulatory impact. So before we can do any sort of fill finish operations, we have to understand what is it that we're filling? What kind of product is it? Okay, whether it's a liquid, it's a powder, or whatever sort of formula product, we have to understand its characteristics. So what are they? And what are those characteristics that directly impact the fill process? So you could have a multitude, even on a liquid level, you could have a multitude of various characteristics that affect that. For example, you could have aqueous, emulsion, suspensions. Even within those products, the type of viscosity. There could be sensitivities around that product, temperature, oxygen, shear. And when I say material, there may be some material sensitivities for some products. Some products may not be able to handle, for example, stainless steel, and has to be in some other inert type of material. So once you have an understanding of what you're filling, the characteristics, the next is, what are we going to put it into? The container closure considerations. And again, there's a lot of formats to take under consideration, and it really depends on the product, the end use, the administration of it, so on and so forth. So these are very important questions because these will drive how the process is defined and the eventual equipment and a validation of what that final product is. And here are some examples. You have glass vials, syringes, ampules, flexible bags, bottles, pretty much anything you would see in a clinical or hospital environment. And then once you have that container, then you obviously have to have some level of closure to seal the product from the outside world, whether you would also have closures such as stoppers, overseals, and ports, and the like. So once you have an understanding of what kind of container closure, the type of product, then the idea is that how do we get that product that we just formulated into that container? And there's various liquid filling technologies out there. So based on the product considerations, you have to ask yourself, what's the best way to do it? You know, keeping in mind of the cost, the regulatory impact, the technology. These are all the factors that come into making that decision. So really, the technology has been tried and true with regards to positive displacement pumps and mechanisms. It's taking, and I'm talking specifically about liquids, it's taking the liquid from one part or one area and putting it into another, which is your container closure, through a rotary piston, rolling diaphragm, peristaltic, and time pressure. And like I was saying, the principles of positive displacement filling are tried and true, and so there's really not a lot of room for improvement. In other words, there's no new technology on the horizon that basically would radicalize the ability to fill that. It's a very well understood sort of technology and process. So this is where we discuss more in how can we improve on other aspects of the process. And it's really in the filling environment with regards to the aseptic process. People are the number one source of contamination in any sort of fill-finish operation. So we have to take measures to exclude sources of microbial and non-viable contamination. And how have we done that? So in the past, we've started off with traditional Class 100 clean rooms where people would be gowned and plated, and there would be continuous clean room monitoring. So you would have people operating or maneuvering within these relatively, I would say, large rooms and being able to fill the product in those rooms. So The introduction of components and equipment into these rooms were done through autoclaves, autoclave pass-throughs, aseptic transfers, and once they get those components in there would be a level aseptic assembly. So as you can see, there's opportunities to improve, reduce risk, and improve compliance. Within the filling environment, there's some risk mitigation steps that we have to take. Aseptic liquid filling is a high contamination risk operation. Why is that? Well, product is exposed to the environment. So you have the product that's actually transiting from your source tank or vessel or whatever it was formulated into your container closure. And sterility assurance is only as good as the robustness of the operation with the highest risk. What I mean by that is the fact that whatever the risk element that you've qualified, and it's only dependent upon how well it's been done to basically assure yourself of a good sterility assurance. So what are ways to mitigate that risk? Obviously, some of the things, as I mentioned earlier, is since people are the number one source of contamination, you isolate people from high-risk operations. You minimize direct operator touch points, and really is you shrink the environmental envelope immediately surrounding the filling operation. What I just mentioned about the traditional Class 100 clean room, you have the product exposed to this volume of environment that in some ways you try to control as best you can, but you want to be able to tighten those controls and be able to monitor that to an even higher level. So the industry has moved from the traditional Class 100 clean rooms to basically creating physical barriers between your filling operation and the people themselves, taking this exclusionary approach, if you will. So through the use of isolators or restricted access barrier systems, or known as RABs. The other thing, too, is how you introduce those components into these closed envelope areas and the materials in and out of the filling envelope is through use of transfer isolators, rapid transfer ports, and airlocks. So again, the idea is taking this volume, this envelope that you have your product exposed to, and shrinking it down to allow better control of that environment. So within the fill finish of the product itself, there's a lot of checks and balances and steps during the fill process that you have to make sure that you want to monitor. Examples are inline fill volume weight checks, oxygen level measurement. Maybe the product is oxygen sensitive and you want to make sure that the oxygen has been purged from the headspace. Stopper detection, making sure your closure is on properly, seated all the way down. You want to also maximize yield and low line loss. A lot of these products oftentimes are very high cost, so you want to have the ability to minimize loss and transfer and fill as much as possible. This is more from an economic standpoint. There's also, these next 3 points are really an automation aspect of allowing of a more consistent and robust approach to making sure that the environmental regions are monitored correctly within the fill zone. Various prep cycles are monitored and made sure they're executed properly. And a lot of reducing the manual interventions and operator handling of your product itself. Again, it goes back to your risk mitigation. And that's really the goal, is minimize the operator touch points as much as possible. So what I was talking about earlier was this shrinking the fill zone, is really creating this envelope around your product. And it's especially true for aseptic products. Now you add in this other mix for cytotoxic or high potent products, or live vaccines, or sort of biological agents, and you want to create this environment that not only do you prevent contamination, but you also want your product from getting out into the environment and exposing the operator. So a lot of the requirements from the aseptic versus containment are very synergistic. So the idea is that they go well hand in hand, and a lot of those principles fall in line with each other. So what are some methods of achieving this sort of ability to protect the operator and the environment? Because up until that point, we've all been focusing on aseptic processing, but now since the products are becoming more and more potent and cytotoxic, what are some of the ways? So one of the biggest movement that we've seen in the past couple of years in the industry is single-use disposable components and products for direct product contact parts. What are some of the advantages of that? It eliminates the need for CIP and SIP, so you don't have to worry about cleaning and autoclaving, all these reusable parts. So that in itself, the qualification is extremely streamlined. You don't have to worry about that and the requalification and the like. Decontamination is less intensive. So you bring in your single-use equipment, you set it up, you're done, you contain it, and then you discard it. And then you move on to the next batch. And a very important aspect is the economy of this. It's very less capital intensive for single-use equipment. You don't have to buy all these stainless steel equipment, all these reusable components that you have to worry about cleaning, setting up, requalifying, so on and so forth. So from a regulatory standpoint, it can only be very streamlined and advantageous in those respects. So I just wanted to just give a brief overview of Biotechnique, which we are a CMO cytotoxic manufacturer, of what we are doing to basically address some of these concerns. We have an integrated Bosch filling line, and we have an inline wing check weighing system, and we are using single-use disposable direct product contact parts. What that means is that all the product that goes through from the supply vessel all the way to the needle itself that dispenses the product into the vial, it's all single use. So at the end of the batch, we could just go ahead and bundle it up and discard it and start from with another set there. What are some other technology improvements? This presentation was really about shrinking that fill region envelope. So let me take you a step back. So we went from the traditional Class 100 clean room, shrinking down to isolators, and now there's actually sort of opportunities to even shrink that further with closed vial filling. And that's where the product does not even see the external environment. Remember how I was mentioning within the isolator itself or the Class 100 clean room, the product actually sees the outside environment to some level. Here, the vial in essence becomes the isolator itself. So what you have is you have a fill needle coming in and penetrating a septum that's already in the vial, placed on a vial, and filling it inside the vial itself. The fill needle is extracted, and that hole that was previously there in the septum is laser sealed. And these vials come pre-sterilized through gamma radiation. So this kind of gives you an idea of what the shrinking the fill envelope is. It's really just down to the vial itself. So here's the summary and really the regulatory impact. The fill principles remain tried and true. What I mean by that is we already have very accurate filling to meet label claim and drug potency. So the opportunities for improvement for risk mitigation is shrinking that fill zone, going from the traditional clean room to closed vials. And what does that really do from a regulatory standpoint? It enhances sterility assurance, mitigate contamination risk, operator and environmental safety, ease of control. I also want to add in here single-use disposable equipment is also very key in this sort of a suite of tools to be able to achieve that goal, is because from an economy standpoint, you only have to use it once, and it's validated once, and there is no reuse, and it becomes very cost-effective at that point. So as a result, you have a new paradigm, has very favorable regulatory impact, the risk profile assessment is streamlined, and the bottom line is really to improve patient safety. And thank you very much, and if you have any questions, please let me know. Yeah, the validation approach would be very similar. Obviously, you would need to do some level of confirmation just to make sure that what you have validated in facility A works in your facility B, C, or D. It's a streamlined level of validation. You don't have to necessarily go through a full-blown qualification, and you can rely on some of that historical data to guide you in what that streamlined validation would be. So,
So does this technology, is it flexible enough to do different sized vials? So this is what is this? Are the vial sizes the same as they are in glass, and does this fill that same capability?
Are you referring to the closed vial technology? Yes. So there are different sizes of vials that you can do, and it depends on, again, what size vial you want. You just have to specify it, and it can actually be made accordingly, and they would have a standard validation package that you would go through to be able to qualify whatever the new format would be.
One thing, just for clarification, the capabilities that you have at Biotechnique, is that, do you have live viral fill capabilities there at this point?
Yes, we do.
So you can do live viruses, and what about other products that are not viral-based, conventional biotherapeutics, for example?
That's a good point, and we're trying to work through from a regulatory standpoint of the sort of the separation that's kind of come to be expected within the industry, that you don't necessarily mix non-cytotoxic or non-highly potent drugs with highly potent cytotoxic live viruses. We're trying to work through some of those issues in terms of getting some regulatory guidance on that and what the expectations are. From a engineering controls, it's relatively straightforward, but it's really how the interpretation of what the regulatory guidance is pointing us that would determine whether we can do that or not.
And what about decontamination then? How does one decontaminate your existing system?
So within our facility, we have a decontamination autoclave. So at the end of the fill batch, we would gather up all the product contact materials, and there is a dedicated decontamination autoclave that we would put this material, and we would use heat sterilization to basically inactivate whether it's a live virus or if it's a biological agent. That's what we would use.
And what about the actual isolator itself, the RAB? How do you decontaminate the isolator system?
So the isolator system, we would do a series of decontamination wipe downs with various, depending on what the product was. If it was a chemical agent, we would use some sort of inactivation solution to be able to wipe down all surfaces and have all surfaces contacted with that. We would also use some other agents to be able to, if it was a live viral or a biological agent, some other methodology to wipe down the areas. But the risk of the agent getting outside of the product train is very low because we would use closed manufacturing processes. They would be contained within the product train itself. So there is the risk of the product getting out into the walls of the filler or the room is very low because we're using single-use disposable equipment. It's all contained within that product train itself.
Any other questions? Yep.
So this is a passive RABS system that we have. When the passive RABS that we have relies on the HVAC within the clean room itself. So in other words, the active RABS typically has its own air conditioning or HVAC system in there, air conditioning system in there. The system that we have does not. It's a RABS with a filler inside the barriers that sits inside an ISO 5 room. So it relies on the room air itself to create the laminar flow through the RABS and on the room itself on the returns back to the system itself. Yeah. So the question is, what was the biggest challenge for us in developing single-use equipment and the manifolds themselves, right? Is that how I understand it? The biggest challenge that we've encountered is really a couple things. One is product compatibility, right? Because a lot of times the single-use disposable equipment is polymer-based, some sort of a plastic. So if you're dealing with, for example, products that are solvent or have some sort of ingredient that is not compatible with single-use, that is an obvious issue. And we have to go find an alternative as such. The other is really how do you integrate the single use on a piece of equipment that was designed for stainless steel, So it can be done, but there's some engineering challenges that has to occur, such as the fitment and the integration and the control of how the single-use disposable set works in concert with your existing system. So those are really the two challenges that I would say.
Thank you, Victor.
Thank you.
Okay, our final speaker will be Debashis Sahoo. He is the Chief Development Officer for madeincell Development LLC, located in Milford, Connecticut, or New Milford, Connecticut, a company that has led in creating unique sterile transfer technology solutions for over 26 years. Madeincell delivers innovative sterile filling, sterile multiple dose dispensing, sterile transfer, and packaging sterilization solutions that answer the toughest challenges faced by the pharmaceutical, biotech, healthcare, nutrition, and cosmetic industries. Debashis joined madeincell after holding various academic and professional positions spanning over 17 years. He has now over 10 years' experience working with the management at madeincell and his global partners and clients in the U.S. and Europe, along with regulatory authorities from those countries as well. Debashis earned his bachelor's in technology degree from Aerospace Engineering Department of IIT Kharagpur, India, in 1998, a master's in science degree in 2000, and his PhD in 2005 from MIT's Aeronautics and Astronautics Department in Cambridge, Massachusetts. Debashis' efforts has resulted in authorship of patents, peer-reviewed papers published, presented, and cross-referenced in several highly respected professional journals, conferences, and symposia. And he's going to be talking about really disruptive technology and some evidence about why it's disruptive and a pathway to transforming the fill-finish industry. Debashis.
Thank you, Susan. Can you hear me? I'll give you an introduction on a new technology that we have developed called closed intact transfer. It's essentially a closed system transfer technology and has applications in filling and multiple dose dispensing, as well as sterile transfer and connection technologies. So as Susan pointed out, what madeincell does is it has bridged the two fields of microbiology and physics to come up with new solutions for ensuring sterile transfers in filling, dispensing, and also in connections. So I'll show you some test cases where we have shown improvement in safety of pharmaceutical and nutrition products, and also an improvement in the therapeutic index, which is basically increase the efficacy while reducing the side effects. So it's a very different approach that we have undertaken to resolve issues that we have seen in the pharmaceutical industries. So this is a snapshot of the history of the company, and the reason why we have this slide is for you to see how the technology has evolved from the beginning. It was founded by Dr. Pai after his experience at Merck, and his background was he was an MD and as well as a PhD, SCD in physics. So now you can imagine bridging the two fields, physics and microbiology. The first generation technology was essentially what Dr. Lee talked about a while back, which is the closed vial filling technology. It was licensed to GlaxoSmithKline in the early 2000s, and the technology was also qualified at the PDA with some media fills. Essentially, what this technology does is you're introducing a pre-sterilized closed vial into the filling equipment, and you're filling from a needle which has open eyes. So the environment around the needle is classified and controlled. So the next interesting application was on the dispensing side. We worked with Nestle to market a new product called VRG. And one of the interesting aspects or a side effect that came out of this work was the improvement in shelf life of a product. So we had a dairy product that we filled about 5 years ago, and when we have visitors come to the company, it's still sterile and it's still fresh, and we keep on dispensing it in front of the visitors. So how did this happen? And one of the hypotheses we have come up with is that from the entire production cycle, from formulation to sterilization to filling and dispensing of the formulation, there was no contact between the product and the environment. So this was a side effect of that. In terms of regulatory milestones, we have had 2 FDA non-objections on the food side. And this particular dispensing technology, we got a new standard from the NSF and this is the first time that we could do multiple dose dispensing of dairy products without requiring refrigeration. And during the same period, the closed vial technology got an approval in Europe for a vaccine. So the last 2 to 3 years, we have focused on a new technology, which is basically you're introducing a closed vial and you're filling through a closed needle. So the needle only opens when it's inside the vial and the product gets transferred. So this is a completely new system because at no point of time is there product contact with the environment. And the same technology is applicable also for the connection system between the formulation vessel and the filling equipment. It's exactly the same technology. So currently we have set up 2 lines in India for a CMO. And we have worked with PATH to do some validation exercises with this technology. And right now, currently, we are working with the FDA both on the food as well as on the pharma side for the qualification. And on the top, you would see the mission of madeincell. Basically, we would like to use this technology to ensure that we can deliver the liquids at the highest safety standard to those in need. It's important that the word those in need because not all of them can afford the highest safety liquids at this point. The second part is prevention of hospital associated infections because one of the side benefits of this technology, closed system transfer, is that we could address the current issue of infections in hospitals, especially at the point of catheters. The CDC data currently states that we have about 30,000 deaths in the US every year because of this. So this is a big mission of the company. And the last item is the pandemic prevention. We'll explain to you how it is possible to fill and to protect 3 billion people in 20 days. It's a new way of looking at this problem. So let's talk about the summary of the technology first. So on the left-hand side, what you see is the standard aseptic filling of open vials in a classified environment. And the environment is protected. The operator is gowned up. So on the right-hand side, what you see is a closed needle, which is open only when it's inside the vial. The product gets transferred, and then the needle closes before being withdrawn. So at no point of time is there any exposure of the product to the environment. As a result, the environment essentially becomes immaterial or non-classified. So we have done some risk analysis on this technology. We have compared the exposures of traditional open vial filling versus this system. And the risk analysis currently states that we have about 1 trillion times less exposure. And in terms of the cost of implementing this technology, this type of line, it's 10 to 20 times less than standard aseptic lines. In terms of regulatory feedback, we have received a non-objection to file ANDA. This happened at the last 2004 meeting with the FDA. And we also have had a senior inspector review at our site. And they have actually prompted us to use our site to do a filing for a product which is on the drug shortage list. And the feedback that we have received are very interesting and encouraging. Paradigm shift and the engineered compliance. And we'll explain why they've talked about engineered compliance. And the application for pandemics is that you could deliver 15 million doses in a day with one of these filling equipment. And because the environment and the controls are engineered into the design of the system, so the filling equipment, setting it up and all, it's very quick process. So the estimate from our supplier is about 3 months from purchase order to installation of the filler. Okay. So this slide gives some videos on the two technologies. On the left-hand side, what you have is an open vial being filled with an with a cannula, and both the environment, the vial is all exposed to the environment during the filling process. And the other important thing to note is that there's a cap. The capping operation happens after the filling is done. So in terms of exposure, you have the container, the cap, the cannula, the capping operation all exposed to the environment, and that negates the need for aseptic procedures. On the right-hand side, what you have is a closed needle which pierces through the septum of a closed vial or a closed pouch or a closed bottle, whatever it is, doesn't matter. And then the needle opens inside, product gets transferred, the needle closes and then withdraws. And what we have done is we have actually engineered the shape, size, and the configuration of the needle and the septum in such a way that we can actually put a high bioburden environment on top of the septum and pierce and shown sterile media fills through that, up to 7 log per ml proof. So this did not come by accident. It came over 26 years of work that was done by the company. So this is where we stand right now. So what this means is that, and the same technology applies not just in the filling head, but if you go upstream, when you're trying to do the connection between the formulation tank and the filler, it's the same thing. If you can imagine, the male side of the connector has the needle, the female side of the connector has the septum, and you just do the connection this way. So what's the implication of this? So you have zero product exposure, and all the compliance that you typically have with aseptic processing, aseptic connections are all engineered now. So that's the basic theory. So then implications in terms of the facilities, 10 to 20 times lower. And as I explained before, it's very easy to build the line to install and qualify. It's very simple. And the same line could be used for vials, bottles, pouches, syringes. It doesn't matter. The most important thing is the septum that needs to be presented to the inside the line. So this is an example of how the line could be set up. Let's say you have prepared your bulk, concentrate, and diluents, and you have these are the connectors that I was talking about, male side and female side. You could mix them, then connect through a surge vessel, and then fill inside the filling equipment. The reason for the surge vessel is that this isolates any pressure fluctuations in the formulation area, in the formulation tanks, and you could have the right dose consistency. And the other important thing to note is, please note the change over time. So typically in a standard aseptic environment, you clean, you sterilize the facility, then you sterilize the filling line and all of that. So we have received estimates of about 8 to 14 hours for this operation. And here we are talking about just doing connections and disconnections of disposable systems. So it's quick. The other important thing to note is that you can now, this is the first time you can now also mix previously sterilized components. So this is important in case you have products which are components which are sensitive to heat that you cannot, like live virus, that you cannot really sterilize. So if you have another way of sterilizing those types of ingredients, you can mix them in a sterile manner with this type of connection system. So what does it mean then in terms of product, environment, and operator? So in terms of the product, there's no exposure of the product to the environment at any point of time. In terms of the environment, there's no splashing, no dripping inside the equipment, so no cleaning, no sterilization required, and no drainage system needed for the equipment. And in terms of the operator, again, because it's all isolated, there's no operator exposure to the product. So we have also demonstrated, I'll show you some slides later, different bioburden that we have tested so far, including phages, which are simulations of viral particles. So that begs the question, how did we come up with the demonstration of this technology? So during this process, we also came up with a new challenge levels to demonstrate the performance of the technology. So the first part was, this is a slide that my macrobiologists love. They put together what does a log mean. And so as you can see, 3 log is a typical sneeze, and 6 log is solid sewage and all of that. So the idea of presenting this information is to bring into perspective of what, when we say 7 log, 6 log, what does it really mean? So that's what we have put together. The second point to note is when we typically do challenge studies, if you are exposing the challenge surfaces to aerosolized environment, there are 2 things which are important. How is the contamination maintained in the environment? And secondly, what is the time of exposure? Because, for example, if you have a 6 log challenge and you have exposed the surfaces to a minute, you actually have only 1.5 log on the surface. So that's why both the maintenance and the duration of exposure are critical for this. So that leads to the risk assessment. So if you look at the time it takes for a surface to get contaminated in ISO 5 all the way to 6 log environments, it takes about 3 months. If you have certain contamination in the environment, for that contamination to be deposited on the surface. It's basically settling down of the bioburden on the surface. And we had presented this data in 2013 at the PDA. So then what we did was, we said instead of worrying about the time of exposure or not, we actually put a dollop of the bioburden right on top of the filling port, and then filled and transferred media through this system. And then did the incubation to demonstrate that even after 7 log CFU per ml contamination, you can still achieve sterile transfers. So the next question is, what does this mean? So if you're talking about 6 log in a 1 meter cube of space of air, then, and if you transfer it to an ml of space, it's there's a 1 million times difference in the contamination level. So we said 7 log CFU per ml, so it's about 1 trillion times, a little bit more than 1 trillion times less exposure. That's how we came up with the number trillion times less exposure. So the other interesting thing that we found out was, remember I said in one of the slides about the 5-year-old dairy product. One criteria that the food industry uses is it's slightly different from the pharmaceutical industry. The technology or the method used to detect the compatibility of the product and the packaging material, the limit of detection is in parts per billion, 0.5 parts per billion, rather than what we normally do in USP is parts per million. So this is another challenge that we had to overcome when we were selecting materials. And this goes to what Dr. Lee was talking about before regarding compatibility of materials. So next, in terms of delivery devices, as I explained to you before, the connection system is the same as the filling system. So you have a needle closed on the left-hand side, and you have the septum material on the right-hand side. So this is the first time you can actually do a connection and disconnection under broth. right? So imagine using these types of devices in a hospital environment to transfer drug products to the patient. The other application is the dispensing application. So in here, what you have is a product in a pouch, let's say, and the pouch is stored inside, let's say, heat-resistant material, packaging material, and you have what we call as a multiple dose syringe on it. And at the tip of the syringe, you have a valve. So the way this valve works is that it pushes the product out, and there's no ingress of anything inside. So no bioburden, nothing can go in. And this is what we have demonstrated at up to 8 log per ml. So if you have this type of valve on this type of syringe, which is connected to the pouch, that enables you to fill and transfer and dispense multiple doses of non-preserved products. So this is an example of how we did the microbiological challenge studies, and on the right-hand side you will see the 5-year-old dispensing test. So the benefit of having this type of pouch and a multiple dose syringe is that imagine you're doing the vaccination campaigns in the field. You could actually take this, let's say you're doing it in Africa or Asia, you could take this on the back of your two-wheeler in your backpack, go down to the village, perform the vaccination campaign for the whole day, come back. You don't need to carry a big refrigerator with you when you're taking vaccine types of products. So this slide is an interesting slide because it shows the different type of bioburden we have tested against this technology, starting from bacillus spores all the way to the phage. So let's go back to the capabilities for pandemic prevention. If you have a 250 ml pouch, and let's say you're injecting 0.5 ml, so it's about 500 doses. With one of these filling equipment, you can fill 30,000 pouches. And with 10 of this equipment, you can fill 3 billion doses. So that's a simple mathematics calculation. And at the point of use, even in a hospital environment or in the field, you could, because of the valve technology, you could dispense multiple doses at the same time. I talked about Therapeutic Index a while back, and so the idea here is to show an eye dispensing device, which has the same valve on it, but the idea here is that the product gets delivered right under the cul-de-sac. That's where it's the most sensitive to accepting the product. And plus, you can, so there's no wastage. You can see the 100-year-old lady, delivering it without any issue. And then the side effects, basically you can fill and dispense preservative-free formulations. So if you add the two effects, it has increased therapeutic index. So the next question was, what were the challenges we have faced so far? We have some interesting graphics or cartoons. This is what the industry told us. Every time we develop something new, we approach the industry, and all they said was, don't interrupt us, it's validated. What we have is validated. So, on the bottom side, what we are saying is, you have high-speed filling lines that are currently out there. And they said, how can you fill through a needle and still achieve the same level of throughput? So there are several ways of doing it. I've just shown you one way, which is you can replicate the number of needles that you need for the fill to match the throughput. But the second point is the throughput could be improved because your changeover between two fills is extremely small. It's quick changeover. And plus, now you can fill multiple doses into one container, so you may not need to fill that many pouches. So, regulatory challenge. So, when we first went to the FDA, said, okay, how do we get this technology validated and approved? The challenge we faced was, let's say, imagine you invent a helicopter and you go to the FAA, okay, I want to land on top of a hospital in emergency. And so they say, okay, you need a runway, you need all the infrastructure around it. So, but the good thing was we worked with the FDA to figure out the right path of getting this technology approved and validated at the regulatory level, and that's what we are currently doing. So this is the conclusion, I guess, of this technology, of this presentation on the upstream filling and for the patient side. So on the upstream side, you can prepare a formulation with the ingredients which have been pre-sterilized before. And with the sterile transfer, you could reduce the changeover from 14 hours to 14 minutes. On the filling side, we could achieve compliance with the existing aseptic regulations. That's what we have demonstrated to the FDA. And the product is never exposed to the environment in production. For the patient, it's the same thing. Product never exposed at the point of use. We have a new standard label for the valve technology, which allows multi-dose dispensing to happen. And we have shown you an example of how the therapeutic index could be improved. So I think that's the last slide. Yeah. Any questions?
Any questions from the audience? Here's Brian with the...
Please show picture where the needle penetrates into the vial.
Yeah.
Full stop.
Yeah. Do you want me to bring that up?
Yep.
Yeah.
Does it work, no?
Yep.
Yeah.
It's very interesting. Magic results always raise questions. Before entering the stopper, this needle left needle, before entering the stopper, in what environment is it? A, B, C?
It's non-classified.
So, contaminated needle penetrates vial.
It could, yeah.
It's okay.
Yeah. You can spit on it.
It brings microorganisms into vials.
Nope. That's the whole engineering of how this happened. So we worked on the needle geometry, the materials of the septum and the septum geometry to ensure that this didn't happen.
It is proved.
Yeah.
Okay. The next question, how is integrity of the stopper becomes absolute after taking away the needle?
So the stopper material is self-closing, right? So after the needle goes out, it's a self-closing stopper material. Then it can be permanently sealed in several ways. So the first generation technology, we used laser to seal the stopper after the filling operation was done.
Operation,
Yeah.
sealing,
Yeah.
sealing. Normally we understand that between stoppering and sealing there must be some clean environment. You do it without clean environment.
I'm sorry, I didn't hear you properly.
He said in between stoppering and sealing, you have to be in a clean environment, but you do it in a not clean, in an uncontrolled environment.
So there's a difference here. So the stoppering happens when the vial is made. So when the vial is made, you have the vial, plastic vial on one side, you have the stopper on another side. That's being made in the molds. They get assembled in the molds, then they get removed, they get sterilized by gamma, they come back to the filling facility where they're loaded into the filler in a non-classified environment.
My question is another.
Yeah.
You have taken away the needle.
Yep.
When needle penetrates the
Yeah.
stopper,
Yeah.
it breaks integrity.
No, it doesn't.
Oh.
That's what we have
It's a
proven.
hole.
No, it's a self-closing
Okay.
material.
How
Yeah.
many experiments prove that everything you tell is
Sure.
billions, trillions?
Right. So we have done, as I said before, we have done microbiological challenge studies where we have actually put the challenge on top of the septum or in a broth and done the transfers. And the other thing that we have done is also media fills, some 34,000 units.
44,000.
34.
34,000. I
units.
remember here in the United States, in the 80s, there was an experiment of 8 million vials fills to prove that isolators give the same sterility assurance level as terminal sterilization. Okay, thank you very much.
Yeah.
Other questions?
How would you confirm the needle is sealed before use, for instance?
Right. So So the needle is treated as a closed container. Same thing as the vial. So when the vial comes into, or the needle kit, it's a kit. That's what we call it. When it comes into the fill finish facility, there's a certificate of assurance that says that these are the tests that have been done. So we go through this typical container closure integrity test to show that when the needle was assembled, it's closed.
Because it uses the product pressure to open.
So once the needle goes in, it's on the back of the upstream of the needle. You have the tubing which goes through a peristaltic pump, and it ends in the connector. The connector is connected to the formulation vessel. Let me show you the system here. So your formulation resides here. This is connected. Then over here, this is connected. So because of the peristaltic pump, you can pull the product into the...
So the pressure from the peristaltic pump is opening the needle after it goes into the...
No. So that's done by the filling equipment. So in the filling equipment, you have a holder where you have installed the needle and the tubing. So that goes in, and the whole it's just a mechanical action of once it goes into it, the needle opens inside the vial.
This reminds me of the interface between the introduction of disposable technologies for bioprocessing at the drug substance side and the ability to actually get the aha of the transformation in biomanufacturing, and all the questions and the evidence and the amount of experimentation that has to be done in order to bring novel technologies to the forefront that are safer and more cost-effective to use, especially in a pandemic response when you're dealing with needing to make 50 million doses in 12 weeks. There isn't that much capacity out there to do that on the filling side that can do it in a timely fashion. So, disruptive technology and novel approaches to it need to be brought to the forefront of the industry if we're going to provide that level of safety to the general population. I'm sorry, there's a question in the back? Did you have a question, sir? Any other questions for Debashis?
Thank you.
Thank you, Debashis. Thank you all for coming. Appreciate your attention. Bye-bye.